Okay, thank you. Thanks, Jess. So yeah, I'm going to be talking about my work on boron isotope analysis by laser ablation multi-collector ICP-MS. So the geochemical signature of marine carbonates is thought to be representative of changes to ocean chemistry, with the boron isotope ratio, which is 11 boron over 10 boron, typically expressed in delta notation, commonly employed as a seawater pH proxy. So it has the utility to understand, among other things, the movement of carbon between reservoirs and to trace biological processes. So laser ablation offers a number of advantages over the more traditional solution-based approaches, in particular being the high spatial resolution of these analyses, and also the high sample throughput because of removing the need for sample digestion and purification procedures. However, the adoption of this technique to boron isotope studies has been hampered by reports of both inaccuracy and imprecision. So I'm going to overview the technical challenges of this approach, and then hopefully get through a few case studies of some examples of applications that we've been putting it to use with. And the importance of all this, well, I'll be focusing on tropical corals today, and with coral reefs being one of the most important ecosystems on the planet, acting as diversity hotspots, offering coastal protection and sustaining important economic activities. They're obviously a key kind of organism to really understand in terms of how they can survive future changes to climate and ocean chemistry. And this method is going to play a key role in our new ERC-funded project called Microns to Reefs that started last year, awarded to Gavin Foster. And this aims to take a multidisciplinary approach to generate a mechanistic understanding of coral skeleton growth, to try and understand how they'll cope better in the future. So our analytical setup that we use in Southampton combines an elemental scientific lasers, new wave research 193 excimer laser ablation system, coupled to a thermoscientific Neptune Plus multi-collector. The methods that we use were presented in this paper up here, published a few years ago in Rapid Communications in Mass Spectrometry. And this plot on this slide just shows a series of analysis of 3 carbonate reference materials with differing boron calcium ratios. So the horizontal bars, these are the solution values, so what we might expect the value to give. And then the individual data points are a repeat analysis of laser ablation. And the key thing to see on this figure is that our results were inaccurate, and also not particularly reproducible between sessions. So we found this accuracy and reproducibility issue that others had sometimes reported. And so decided to look into it to try and work out why it was there and how we can get around it. And well, the inaccuracy is proportional to the boron calcium ratio, which is seen on this upper plot here. So inaccuracy on the y-axis, boron calcium ratio on the x-axis, and that's showing the analysis of 5 different carbonate reference materials. So this relationship seems to follow log law, and having already ruled out factors such as plasma loading and laser-induced effects, it became apparent that we had some kind of interference. So the lower plot shows a mass scan across the boron isotope range. And what's key here is that the 3 plots that are using the Faraday cup detectors are showing this raised and irregular baseline or background signal across the entire mass range. So this is implying that we do have some kind of interference. Now, the signal of these backgrounds is proportional to the calcium within the material being ablated. So this top grey line, that's a carbonate reference material. The other 2, one is a silica and one is just a gas blank. So it seems to relate to calcium. Now, obviously, we're not in the calcium mass range, but this can be explained. The light blue plot on this figure is the gas blank, again, but this time using an SEM with an RPQ, which is a type of energy filter, so instead of the Faraday cups. And so this implies that the matrix interference is coming from scattered ions with disturbed energies. So that seemed to be the issue, at least with our setup. And I set up an approach that utilizes this relationship to correct for this matrix interference. And we get sort of accuracies and precisions to about 0.6 per mil. Now, this doesn't explain why some studies report no matrix interference, but this can be explained by it being a model-specific issue, so relating to the design or geometry of the mass spectrometer, something like a flight tube. And on this plot, it's another series of mass scans. The one from the previous figure is this grey one, so this is from the Neptune. And then I've added two others on here. One, this upper one is in blue. This is from a New Instruments Sapphire multi-collector. This bottom one, the red one, is from a Thermo Scientific Neoma. And the key thing here is that they're essentially flat. You don't see this irregularity. And also they don't change with the sample materials. So carbonates versus silicates, they kind of overlap. So it looks like it's a model-specific issue. And so with these, I suspect the key sort of tuning parameters are plasma conditions. But if someone is using an instrument that has the kind of matrix interference issue like the Neptune, then this can be corrected for using the approach developed in that methods paper. So since this investigation, we've begun using our methods in a series of studies. First one I'm going to briefly talk about shows how analysis at high spatial resolution can provide new insights into coral calcification strategies. For this study, a sample of coral Siderastria siderea from the Belize portion of the Mesoamerican Barrier Reef was sampled. And an area 4 by 5 millimeters was mapped for boron isotopes and trace element composition. In the middle two photos, you can see it mounted just for the laser ablation analysis. It's just a 0.5 centimeter thick slice mounted on a glass slide. And then in the image above that, there's so the area that was mapped is in the center of this image, and it's similar growth to, or the same growth as this little area here, which was mapped through solution approaches with micro drilling. So we had a good control on accuracy. And it was mapped by just analyzing 30 parallel transects on the coral. So this was with a 140 micron laser beam. And that was sort of including sample preparation, maybe 2 days of work. To give you some kind of comparison, that would be a few weeks of work if you were to try and do it by solution. But the resolution would also be very different. So the line transects were mapped in R, and this is an image of the boron isotope map for this sample. So the laser approach shows seasonal variation in this direction. So the coral is sort of growing up the slide. You can see a low in the boron isotopes around here on the map. And the amplitudes of this variation were consistent between laser ablation transects, so about 3.4 per mil. However, there's also structural variation going sort of laterally across the page. You can see this band of lower values coming up like this, surrounded by higher in yellow. And what's interesting is the mean values of the individual transects vary from around about 21 to 24 per mil. And they have a difference of about 3.3 per mil. So that's very similar variation as you see in the seasonal amplitudes as well. And this structural relationship can be reinforced when we overlay it with a SEM image of where we've analyzed. So the lower values are in this more porous portion of the coral, so in the columella, and the higher values are in the denser skeleton areas in the thecal walls. And we can calculate the pH from this data, which is shown here in this map. And again, you can see you've got these low values in the center of the polyp, high values in the theca walls. So it's indicating that the aragonite in the theca wall formed in a more heavily modified environment relative to ambient seawater than the central polyp. So the key implication is that the skeleton does not precipitate from spatially homogeneous fluids. And different parts of the coral might have different sensitivities to environmental stress. So the second case study really demonstrates how well this approach is for sampling large data sets or large sample sets, and also for targeting very specific points of growth. So it's part of a master's student project by Mohammed Saeed a couple of years ago, and we're currently writing this up for publication. But the premise of the study is that nutrients, including nitrogen and phosphorus, are key to coral holobiont physiology and play important roles in processes such as skeletal and tissue growth. But a number of natural environmental factors can change the nutrient budget available to these corals. And sometimes this leads to nutrient enrichment and even eutrophication. So whilst we know that imbalanced enrichment of nutrients can have detrimental effects on coral physiology, we don't really know how this affects the mechanisms behind biocalcification, or how they might affect some of the key geochemical properties that we use. So for this study, 32 corals were grown in the Coral Reef Lab at Southampton. So these are Aquapora, Polystoma, and varieties lichen, and they were grown in 4 different treatments. So one is a replete, sort of normal conditions, H and HP here, one with low nitrogen, low phosphorus, and then 2 with imbalanced enrichment, high nitrogen, low phosphorus, low nitrogen, high phosphorus. And these were, after growth, they were sampled, mounted in resin, polished up, ready for analysis. So there were clear physical effects on these corals. Zooxanthellae density was affected in all treatments, shown on this figure on the left. And also skeletal growth was affected as well, particularly in the Aquapora, the branching coral. So they are disrupting the coral calcification to different degrees, these treatments. The top plot here shows the mean boron isotope values for each treatment. We've got parietes on the left, Aquapora on the right, and this is expressed as delta notation, so it's essentially offset from the treatment tank, the cultural waters. And the relative differences between the treatments are fairly consistent, with the imbalanced nutrient tanks showing generally a larger difference in boron isotopes. And this is reflected in the calculated pH shown on this figure down here. And so imbalanced nutrients are showing a greater upregulation in the extracellular calcifying medium of the corals. And they are affecting the boron isotope proxy. And we can also plot the boron isotope versus the calculated carbonate ion. So now on the y-axis of this plot, and see how the imbalanced nutrient treatments have lower carbonate ions in the ECM. So corals balance, they balance their proton and bicarbonate pumps as well as skeletal extension to manipulate their internal carbonate system, optimizing survival. And this is probably the behavior we're seeing here. So it's sort of dragging it down to the bottom right of this plot. And finally, the third case study, which we're still collecting data for, is really good at demonstrating how the laser ablation approach can reconstruct the paleo-environment to even higher resolution. So in this study, we went to a well-studied area, Hog Reef, which is a rim reef on the edge of the Bermuda carbonate platform, and where a NOAA buoy has been moored there continuously collecting environmental data. And previous studies have shown that temperature is the primary driver of coral calcification here. And this is seen in this figure here. This very bottom plot is the net ecosystem calcification, and that is consistent with the temperature log vector here. But in a similar fashion to the Belize samples, they were cored, and we mounted them like this in the laser ablation cell, and small regions were mapped for areas that match the period where the logger data has been collecting. And this figure here shows a couple of maps, say for these 2 cores, 3B and 1B, we've got boron isotopes on the top and boron calcium on the bottom. And it's apparent that there are seasonal cycles in both, with corresponding highs shown in yellows or oranges on here. So we can reconstruct the coral paleo-environment at high temperature resolutions using this approach quite nicely. And using an age model, we can plot this alongside the logger data. And it's clear that this approach will show high resolution and speedy environmental reconstructions that could be used for sort of historic, looking at historic trends in ocean acidification. So I think probably stop talking on this one now, because just to sum things up. So just to summarize, there's a matrix interference from scattered calcium ions that hampers accuracy and reproducibility on some models of multi-collector when using laser ablation, but these can be overcome, and the approach does offer really good benefits for studies of marine carbonates. And I shall leave it there.
Brilliant, thanks, Chris. So I'm not seeing any questions in the chat yet. So a reminder to put those in there if you have them. I have a question. So this is focused on corals. As a forearm person, I'm wondering how low can you go at the moment with your boron calcium?
Yeah, so we've working with Talia Babila, we've been working on some benthic forearm data sets, and she's currently writing up a study on those. And so these are sort of around the mid-range, I guess, because they're benthics, well, maybe 150 to 200 micromoles per mole. Apologies if I just got that wrong, Tally. We've not really got down to sort of the planktic levels of boron concentrations yet, but we have had a go, and I think it's more to do with the samples that we've been analysing. So in theory, they should be fine because we can get accurate data for standards at sort of 50 micromoles per mole boron calcium, but the samples haven't been able to hold up in the laser cell to the ablation. So that's something I think should be possible, but we've not got down to those levels yet.
Thanks, Chris. And there's a question from Joe Stewart in the chat. He says, fascinating that dissolved seawater nutrients can impact skeletal 11B when nutrients can often be derived from particulate organic carbon. Were there fish in the tanks providing a fresh source of food, fish poop, and would we expect the same results in the wild?
To be honest, I don't know about the fish tanks for this specific experiment. I know some of them do in the coral reef lab, but I wasn't part of that side of the project. As for the second part, would we expect similar in the wild? Well, I guess that's something I have to think about, I think.